questions. So, I'm delighted to introduce the first speaker, which is Michael Hayden, who is basically the boss or CEO of Prilenia. Thank you, Michael.
Thank you, Sarah. And I must say it's so wonderful to be here again, to see old friends and colleagues. And I was particularly moved by Nora's presentation last night, and Marjorie has remained a source of great inspiration as we search for new treatments, but also to provide care along the way. So I'm going to share with you a little bit, a quick update on where we are on the PROOF study, which is a predopidine study on the outcome and function in Huntington disease. Now, of course, everybody is fully aware of all the different components of Huntington disease. What we're focusing in Prilenia is on this aspect: manifest patients, stage 3 by the ISS scale, and patients here who've had some drop in TFC. Of course, there are many other symptoms, and we took this drug to approval for treatment of chorea, which is a presynaptic dopamine releaser, similar to Ingrezza. But today we're going to talk about a drug that has impact, potentially, on the maintenance of total functional capacity. So this is a drug that's an investigation drug in use for HD and ALS. It's safe and tolerable. It's given orally. The drug has shown some early evidence, but not proven yet, for total functional capacity. We understand the mechanism of action, which is a potent and sigma-1 receptor agonist, and there is some early data from open label that suggests that the effect may be long-standing. I'm, of course, a big fan of oral small molecules for CNS diseases, by virtue of the oral bioavailability, the administration, clear CNS penetration and access to intracellular targets, and of course, a short half-life when treatment needs to be discontinued. We know that this is initially thought to be a dopamine modulator. We now know at the therapeutically active doses that this interacts directly with the sigma-1 receptor. This was using data, degenerating data using a sigma-1 receptor ligand that binds sigma-1. You pre-treat with your drug and you see how much the binding of fluspidine is decreased. You can see a dramatic decrease. So it occupies sigma-1 and blocks the radioactive label binder. Sigma-1 is an interesting and provocative target for the treatment of Huntington disease. It's highly expressed in regions of the brain that are critical. It plays a role in regulating crucial pathways, and certainly preclinical data shows that the loss of sigma-1 can increase mutant Huntington aggregates. It's located on the mitochondrial-associated membrane, and predopidine interrogates the target at that point. With that, it also has impact on numerous organelles in the cell. Mitochondrial function is significantly improved. It also enhances autophagy, increases BDNF, has significant impact on rescuing and providing homeostasis for the ER, and also is important in nuclear cytoplasmic transport and nuclear pore stabilization. With all of this, this is associated with enhanced neuroprotection. Lots of data, and of course, how do we know that this is truly mediated through a single gene? All these experiments are done, and then these animals or cells the sigma-1 receptor is deleted, and what we learn from that is the effect, the neuroprotective effect, is completely ablated. This is work from Chris Ross's lab, showing just cell death from striatal neurons of different animals. Purple here, showing increased cell death. Predopidine, showing that you can restore viability in this particular model. If you cross those animals to a sigma-1 receptor knockout, you lose the effect completely. And that effect has been replicated in about 30 labs by different investigators around the world. Here you can see spines, of course, decreased in Huntington's. This is a YAC128 model, much less spines that you can see here. Predopidine restores spines, and again, in the absence of sigma-1, that effect is completely lost. And a paper just out 2 weeks ago from Fred Sardou in collaboration with us actually made the cover of Neurobiology of Disease, showing that pridopidine actually rescues BDNF signaling and restores the number of striatal spines in this beautiful model that Fred spoke about yesterday. So all of these, and there are numerous others, to show how pridopidine might be working. We also had some data from PRIDE. And of course in PRIDE, pridopidine failed to reach significance for its primary endpoint due to primarily an unexpected placebo response. Here's the data. Here was PRIDE showing improvement in TMS, but there was a massive placebo response that went out for 52 weeks, and there was no placebo subtracted significance associated with this, very different to MERMAID, which actually showed some significance in prior studies. Total functional capacity is a well-recognized endpoint, validated and accepted by the regulators. For families, this is a key issue. How do they continue? It measures those things that matter: capacity to work, household activities, managing finances, eating, dressing, and taking care of themselves, all of which are so important in terms of maintenance. And it's measured using the TFC, which is looks at occupation, finances, domestic chores, activity, daily living, and care level. And this is endorsed by both the FDA, which has said this is an acceptable single primary endpoint in phase 3 from the minutes. Also, Beam Farm in particular acknowledges the meaningfulness and acceptability of TFC. So it's endorsed by both, it's been used for 4 decades, and has widespread regulatory acceptance. We showed already in PRIDE in all patients, which was a pre-specified exploratory endpoint, that these patients on 45 milligram BID showed less progression based on TFC compared to placebo. We also see that this is a drug that actually was the first to show some maintenance of TFC compared to a whole host of other studies. We've also looked for other evidence post hoc. NFL, this study was started in 2013. NFL was not a marker at that particular time, but we had some blood available from patients. The SOM methodology was used, and these were analyzed in the usual way. And what you can see here, firstly, is there's a significant correlation between delta TFC. This is in placebo and NFL, and this remains significant after correction for numerous. So as your TFC essentially got worse, there was an increase in NFL. And then also when you look here, this follows as expected. PRIDE was very similar to what's seen in TRACK-HD and modeled NFL from other studies. And the NFL with pridopidine was maintained or stabilized. And this also gave further understanding. And these are the 95% confidence limits. And you can see with 45 milligram BID, you didn't go very high. And there was really a significant difference with that seen in placebo. Another important thing, the drug is safe. The drug is safe and tolerable, easy to administer. And that's also, of course, such an important issue. So our phase 3 trial is designed to replicate and assess the effect seen in PRIDE in a much larger sample. And these are the baseline characteristics. We have 499 patients, very similar to PRIDE. The age is similar, weight, CAG length pretty much the same, and a similar proportion of males to females. So baseline demographics are very similar. What is different, here was PRIDE. We have in PRIDE the earliest stage of Huntington disease based on TFC was 29%. We have slightly earlier HD1, which again will give us some indication of looking at the effects of this, particularly in early HD. It's been granted fast-track designation by the FDA, which has allowed us to already have some more frequent meetings. We've had more written communications. And on results, this will be eligible priority review and also a rolling NDA that's already being prepared. These are the investigators. This has been done with the Huntington Study Group. These are the PIs and the co-PIs, many of whom are in this audience, and this has been formally endorsed by EHDN as the trial. This is the study, just to go through the mean change. The endpoint is the mean change from baseline in TFC. There's a whole host of other. It's 65 weeks, and the population is really early HD. What is interesting, this study started during the midst of COVID. We were very worried that, in fact, we'd have diminished recruitment because of this. But in fact, what happened is that we enrolled 6 weeks ahead of schedule and above target. First patient was randomized almost 2 years ago in 2020. And in July, just recently, there were no safety signals of concern, and the study could continue as planned. Here we are. And what's also remarkable is that there's a very low dropout rate, about 6-point-something%, so very low. And all patients who are eligible have now gone into open label extension. In conclusion, I just want to say this trial is designed to test the hypothesis that pridopidine may have impact on total functional capacity. But it's important to be sober about the prospects. Just to remember, phase 3 outcomes in neurology and rare diseases, 40% fail, 60% are successful, and we really know going into this that this is not a sure bet. We have to manage expectations as we look to getting the results. And these results will come available around the second quarter of 2023, early 23. And so we're hopeful, look forward to this. This would be important if we can have any impact on some aspects of maintaining function for Huntington disease. And I want to thank so many investigators here, the lay organizations and the HD community, the participants and their families for their trust and ongoing collaboration. And we're just waiting to see what happens. Thank you all.
Thank you. That was perfect timing, Michael. Thank you. Time for one question, and then write your questions down for the panel discussion so we can have lots more questions for Michael. Any questions? Is there a single one-off question? Come on, guys. Question over here. Question over here. Put your hand up. Yeah. Can you introduce?
How often? Yes, Herwig Lang from Düsseldorf. How often is the Data Safety and Monitoring Board checking on the data?
The unblinded data, and at the moment it's looking pretty good.
Thank you, Herwig. Okay, write down all your questions for the panel discussion.